Okay, once again, hello and welcome to everyone to today's webinar. Actually, today we have the first webinar of a series of webinars with the title Fundamentals and Application of Stable Isotope Labeling in Proteomics and Metabolomics. And we are very happy to welcome Dr. Markus Krüger today. Hello, Markus. Dr. Krüger is from...
You're frozen now, but at least I know it's better.
Dr. Krüger is from the Cologne Excellence Cluster of on Aging and Aging-Associated Diseases, and we'll start the series of webinars today with the title Proteomics using SILAC, Applications in Cell Culture and Living Animals. So before I give the floor to you, Dr. Krüger, I would like to mention two organizational points. First thing is, the chat function is on. So whenever you have a question, please feel free to use the chat box. You can find it on the top menu. And if you click on chat, on the right-hand side, window will open. And please feel free, whenever you have a question, post your question here, and I will read it to Dr. Krüger. But you will also have the opportunity after our webinar in a question and answer session to activate your camera and to activate your mic and to pose your question live to Dr. Krüger. The second thing is, after this webinar, we will have two more webinars. The first webinar will be directly after this webinar, presented by Dr. Jürgen Cox with the title Multiplexing with Isotopic Labels for DDA and DIA in Max Quant. And the second webinar will be today in a week at the same time, presented by Professor Dr. Christoph Turk with the title Stable Isotope Labeling in Mammals, a resourceful method for protein quantification and beyond. So after this meeting, I will post the registration links to both other webinars in the chat box. And with this, Dr. Krüger, I would like to give the floor to you. I am very happy to learn today.
Okay, thanks, Marcel, for the nice introduction.
So while preparing, we will have between 35 and 45 minutes of presentation. And after the presentation, you will have approximately 15 minutes where you can live pose your questions to Dr. Krüger.
Okay, now it's perfect. Okay, so that is vice versa for me, but doesn't matter. Okay, so wait a second and go for the laser here. Good, yeah, sorry again for the confusion here with so Corona is quite a while, and so we're not used to anymore to this technique here. So as Marcel said, right, I mean, whenever you have a question, just post it. We might also make a short break or a short question and answer time in between if necessary, because I have one part for the basically in vitro labeling or the typical SILAC approach, and then the second part I would like to introduce a bit more SILAC labeling in vivo. Okay, so that's our institute here. As Marcel said already, I'm in Cologne in Germany, and we are quite interested in aging-associated disease, and I'm working a bit on skeletal muscle tissue and heart tissue and different disease entities. Okay, so just to present a bit the okay, just to show you a bit the overall workflow, what we like to do with our samples, right, and also SILAC labeled samples. So what we start usually, and you might do the same, you have your cells and you lyse your cells or tissues. So then we digest and we extract the proteins in a certain buffer. So then we digest our protein with specific proteases. Then we have the option to enrich post-translational modification like phosphorylation, acetylation, deglycine remnants. It could be any modification or cellular compartment, whatever you like. And then we're using our LCMS system and usually using either the quadrupole orbit trap instruments or the ion mobility instruments basically to measure our peptides. And then later on, we have some software tools like Max Quant, and I'm sure that Jürgen will introduce a bit more into this topic and this software tool. Diane and Spectronaut are other programs which allows you to analyze your data. And of course, I will not touch it today, but I mean, so what we could do is, or what we can do is, we have different acquisition methods, how we measure our peptides. And if you are familiar with the mass spec workflow, you might know that there is either the data-dependent acquisition mode, where we select specific peptides and fragment them, or we basically fragment a whole window of intact peptides into smaller pieces in this fragment 1 here, and then later on we analyze these mixed MS spectra and try to extract single peptide intensities. And for what is good for, so we like to, and not we, but also other people like to use, of course, global expression proteomics or targeted approaches where we focus on specific candidates and proteins, or a protein-protein interaction map, so to see what proteins interact to each other, right? Those things can be done here, or of course the analysis of post-translational modification. Yeah. Okay, and now of course one major question is how we quantify and how we identify those proteins and peptides, and quantification mainly. So then one idea is of course to using this so-called spectral count. So what we do is that we count those spectra which are identified in a healthy situation, for example, here in sample A, and those spectra which belongs to a specific protein in sample B, which might be disease condition. And then we counting these spectra, and just by simple protein, by counting those peptides, we get the quantification of the proteins. An alternative is the ion intensity-based quantification, and this is actually what we use for label-free protein quantification, which is the main tool or the main quantification method what a lot of people are doing in our days. And here we're using the intensity of those peptides, or in the DIA mode of the MS spectra, and we're comparing basically the intensity of the peaks in the control run and in the second run in the sample B. The problem was, and then of course we can quantify the intensity from the green peak and from the red peak. The problem in the old days was that of course you have to measure a lot of samples and comparing all peptides to each other. Yeah? And that was kind of an overload for the computing power, and was a major effort. And for that reason, people basically or labs and developed these so-called heavy and light labeled proteome, and they call it either on a chemical way, ICAD or tandem mass spec, or if you want to use a metabolic labeling strategy, stable isotope labeling by amino acids in cell culture. And this chemical labeling was mainly invented by Rudi Ebersold in almost more than 20 years ago, and a bit later, so then this SILAC labeling came into the game. And the idea of both techniques, of the chemical labeling and the metabolic labeling, is that we generate a state A, a light state, which we already have, and a heavy state, right? And then we could then mix both together, which you would never do if you do Western blot analysis, right? I mean, there you have always two samples, you load them differently or separately on the gel, and then you look for intensity of a band. So what we do now is we mixing those two conditions together, and then we could enrich, purify, or do whatever, and then use the trypsin digestion to get peptides and measure them with LCMS. And the result is, of course, that we get a peak, right? A light peak and a heavy peak, which is separated by a certain mass range here, right? So this allows us the relative peptide. So with other words, we don't have to compare now different runs to each other. So we have two samples, and we can merge those samples and then compare them directly. Yeah? So that was really a major step forward in terms of accuracy and reliability during the days when SILAC was invented. And how is it working? So the idea is today to give you more basic knowledge about those techniques, I think. So what we could do is we could then take certain amino acid, and of course the best amino acid, and I'll come later to that, are arginine lysine. So here's an example for the lysine, which is here the light lysine. Yeah, I mean, this is a normal structure here with all natural isotopes. However, if we could then exchange these carbon atoms from C12 to C13, so then we have 1, 2, 3, 4, 5, 6 carbon atoms, and our lysine would be then 6 Dalton heavier. Yeah? And the same is true for this lysine 8, where we introduce 2 times the N15 instead of N14. Yeah? So meaning we could then have an 8 Dalton difference compared to the light lysine. Yeah? So that's the idea here. And how is it working then in cell culture? Yeah, because this is mainly used in cell culture, or was used in cell culture. So we have our light cells here shown in green, yeah, and here. And what we do is now we spike in either the lysine 0 or the lysine 6. And then we, after 5 cell doubling, sub-doubling, so meaning you have to split your cells, depends a bit how fast they grow, but usually we say you split your cells 2 or 3 times. And then you get a mass shift of 6 Dalton, since we have spiked in lysine 6 here. That could be different for different amino acid, but here we would then have a 6 Dalton shift. So the green peak might then or will disappear, and then we have basically a 6 Dalton heavier peptide peak. And this is true for all peptides which are labeled with lysine 6. And then, of course, what we do is, as I said, we mix these two samples together, and then in case we have equal peptide and protein concentrations in these two samples, so then we would have a 1-to-1 mix, right? However, if there is something wrong with a certain peptide in the heavy proteome, we might see a reduced intensity of that peptide or an increased intensity, right? So this is always a relative quantification. Yeah, good. So that's basically the idea of SILAC, and that's actually it. I mean, that's, if you have understand it, that system, then you might do all this experiment and then use this technique for you. Okay, so and why is SILAC on trypsin digestion a perfect match? I mean, this was also a quite nice idea at the beginning from Xiaoyan Ong and Matthias Mann, of course, is when you use arginine and lysine, right, which are two essential amino acids, so then all proteins are labeled either, again, as I said, with lysine or with arginine, right? And digestion of trypsin results in lysine or arginine containing peptides. So with other words, if you use these two amino acids as SILAC amino acid, then all the peptides which originate after trypsin digestion are labeled and can be quantified, right? Of course, if you only label your proteome with lysine and you use then trypsin digestion, then you are blind for the arginine containing peptides. So they are not labeled, and then they overlap to each other, right? So that's the idea. Okay, so that's basically the idea. What we can do is, and just some brief setups here. Of course, what you need medium, DMEM or RPMI or any other medium without the natural isotopes for arginine and lysine, right? They should be depleted for that in both conditions, in the light and in the heavy future condition. And then you need also dialyzed serum, which is depleted for free amino acid, because this would then, if you use normal serum, this might then contaminate your samples. And you spike in then arginine and lysine, right? So this is what you basically give to your media, and you use either light lysine or heavy lysine or arginine, and then you have your ready to go your media, and then you basically label your cells. Other amino like methionine can also be used, or leucine, so these are also available in cases. So for methionine, for example, is just one example here, this heavy methyl SILAC labeling. So the idea is not to label the proteins, but instead you give this heavy methionine here, and this heavy methionine or methionine is then converted to S-adenosyl methionine, and this can be then tracked and monitored by protein or DNA methylation, right? And this is also an idea how to track protein methylation and quantify these things. Okay, so the problem is, as I said, I mean, we have two conditions, light and heavy, or state A and state B, but very often you have more conditions, right? And this is, for example, the idea that TT can go up to 32 plexing now nowadays. However, I mean, this might be a disadvantage of SILAC, that you cannot go up to 32, although there is other techniques like null code, which might be an option to increase the multiplexing. However, today I just want to say, okay, we have state A and state B, and we have also state C. So why is that? So we could use basically our non-labeled condition, of course. We have the middle condition and we have the heavy condition, right? So this is either with arginine 6 or lysine 8 or lysine 6, and we have arginine 10 and we have lysine 8. So these are the three conditions what you could use. And what we basically do then is, and these are the pairs which you might expect after your LCMS analysis, and these are doubly charged ions here for arginine or for lysine, and this is basically that we divide the masses by 2, right? So arginine 6 will appear as a pair with 3 Dalton mass shift, and arginine-10 will appear as a pair with 10 Dalton mass shift, right? Or 4 here. So, and of course, the same is true for the triple labeling. You divide your SILAC labels by 3. However, some people use also the combination of lysine 0, D4, and lysine 6. The problem here is if you use lysine 6 as the heavy amino acids, which might be okay. However, if we have only 1 Dalton difference here, at the end, because our peaks here appear not as a single peak, but what they appear as a kind of an isotopic envelope, right? As an isotopic pattern. So usually we have 3 peaks per peptide. And once we have basically here lysine 4 and lysine 6 with 1 Dalton difference, so then it might be that the peak, the third peak of this lysine D4 will overlap with the lysine 6 peak. And this, of course, will basically give you strong positive quantifications, right? Because you have an increased signal here. Therefore, this combination should not be used. So this is basically the method of choice or the combination of choice for a double triple SILAC experiment, right? When labeling with arginine and lysine and have 3 conditions. Okay, 3 conditions. How do you get that usually? So if you want to basically increase the multiplexing, you combine 2 of these triple SILAC experiments, right? So, and this is just one beispiel, what I did a couple of years ago when I started my lab experience with SILAC. So what I actually did is here, so because we had 5 conditions, right? Or we want to analyze 5 conditions. And these conditions are here treatment of something, doesn't matter. For no treatment, then we have 5 minutes, we have 10 minutes, we have another minute here, and we have 20 minutes, right? And you already see that the 5 minutes time point shows up 2 times. So what I did is basically I mixed up these light, middle, and heavy, right? And then I have basically a mixture here, and for the second experiment. However, before I used the 5-minute time points for a common standard, right? So these 5-minute time point between these 2 experiments were actually the same. So what I did is I mixed up the 5-minute as a common pool for both experiments, and then I spiked in this heavy pool from both experiments into my light and 10-minute treated samples, and equally in the 1-minute treated samples and 20-minute treated samples. That could be any other experiment, whatever you like, right? And then, of course, we measured these 2 samples individually, and then we overlap basically the intensity of these peaks because the 5-minute is the common peak, and this allows us to directly compare these intensities to each other based on SILAC quantification. And then we can go up to, in that case, 5 time points. So we have 6 experiments or 6 plates, and we're using double one plate, so meaning we have 5 time points to analyze. So that's the idea, to increase the multiplexing or to basically get up to 5 conditions once we're using these experiments in combination. And of course, one can also, let's say, overdo it or increase it, right? So Henrik Neut in my lab, so he basically now wanted not only compare 2 double triple SILAC experiment, but he basically combined 3 of those experiments, right? And that's, or what we can also do, you can even combine 5 of those experiments. So this is also possible. So 4 of those experiments. And this gives you just an example here. If you basically go for an unbased screen for kinase activity, so what we did is we stimulated our cells with insulin, as I said. Here, this is a 10-minute insulin treatment here in that case, also in that case. So these are all the same conditions, and in that case. So we have 3 SILAC experiments. So this is one triple SILAC experiment. This is one triple SILAC experiment, and this is one experiment here. So, and then we basically had treated, we have the basal condition where we see no phosphorylation of ERK because insulin stimulate not only the insulin receptor, but also the MAP kinase, so phosphor-ERK, and we get basically activation. Here are open 10-minute insulin stimulation. However, if we now basically treat these insulin-stimulated cells with this GSK3 inhibitor, we don't see any change, which is known. However, if we now stimulate the cells with the MAP kinase inhibitor, we see a decreased intensity, based on the SILAC intensity of the light peak. And of course, the same is true for the other inhibitors here, which do not show any decrease stimulation. So therefore, you might then basically compare these basal condition plus insulin stimulation and the different inhibitors. You might also use other, of course, treatments. So that's the idea, to directly compare all these treatments. And of course, as I said, you can also compare 1, 2, 3, 4 experiments together, right, and have these 10-minute insulin stimulation as a common standard. And here I don't show all the spectra, but what we do, or what we like to do, is we use then this color code for these SILAC ratios, and then those sites which are affected by the treatment, by these different treatments. Right? In that case, we have again a decreased phosphorylation of the MAP kinase once we use this MAP kinase siRNA here, or the inhibitor here. Okay, so that's basically the idea of combining different SILAC movement to increase the multiplexing. Okay, so of course, I mean, this is kind of multiplexing, but then the question might be, how can we also use this SILAC technology to quantify human tissue or any other tissue? And one idea was again invented by the Matthias Manns lab, that one used basically different labeled cell culture models, and they called it super SILAC. So what they did is actually, or what one can do is, you use different kind of lung cancer cells, whatever that is, so 3 different cell culture lines, or even 4 or 5, to basically, and label them all with these lysine 8 and arginine 10, so make them heavy. And then in the next step, you mix them all together to get a kind of a super SILAC mix. So the idea behind that is that you increase the proteome, or the peptides and proteins, which are basically heavy SILAC labeled, to get a wide proteome, which then, if you know, and this is just a control, that you have a heavy proteome of 3 different cell lines, and then what you could do is, you could then spike these heavy labeled proteome from cell culture into the patient lung carcinomas, right, in different patients. And then, of course, you have always the same standard, and you hope, since you have already 3 or 4 or 5 labeled lung cancer cell lines, that you cover all the peptides and proteins which are in all these different patients. So that's basically one idea to quantify human tissue with a kind of a super SILAC mix. However, I would say honestly, I mean, what I learned now, or what is a tendency, is that also for those patients here, one could then use also the label-free approach instead of using the super SILAC. But however, I mean, this is again definitely an alternative to get accurate protein quantification. Okay, as a first summary, just for this in vitro part, of course, label-free approaches are probably the method of choice for global proteomics studies. However, SILAC is still a versatile tool and a useful tool if you want to go for PTMs or for fractionations. And just as a summary for you, as a take-home message here, is that basically you have to label your cells for 3 or 2 to 5 cell doublings. It depends a bit on your cell culture model. You should, of course, check the incorporation by a short measurement that everything is heavy labeled, and then you mix up the light and the heavy labeled proteins 1 to 1, and then you might enrich phosphopeptides, right? Or you fractionate your cells and then do the LCMS analysis. Right. So one point, what you should be aware is that sometimes, I mean, it might happen that some cells, they might convert arginine to proline. They haven't touched it today, but this arginine to proline conversion could be a problem. So what we do, and should be checked, but what we usually do is that we adapt or reduce the arginine concentration. You can also increase the proline concentration, but that might definitely help to use a bit, reduce the arginine. And and of course, some cells, and this is also a cons or a point which one have to discuss, is that of course some primary cells, they don't like to grow in the same SILAC media, not because of the amino acid, but as I said, we have a dialyzed media, right? So everything which is smaller than 10 kD or 3 kD, depends a bit, is depleted, and of course a lot of growth factors are also depleted, and this might then inhibit the growth of those cells. You might add some growth factors or other supplements which might help here, right? So again, SILAC and TMT labeling whatever you like, is definitely a useful and accurate quantification method for PTMs, where you have only one peptide to quantify, and of course, if you go for several step on protein fractionation experiments. Okay, so that was the first part. I don't know, Marcel, are there urgent questions for that first part, or should I just go on?
Everything was so clear that there are no questions so far, but
we can also discuss it at the end. Yeah, it's fine. Yeah.
Okay.
Good. So then we're coming now to the second part, right? So I mean, of course, SILAC is a nice tool in cell culture. However, I mean, you might also use for human tissue or for mouse tissue. However, I mean, even in those tissues like erythrocytes or kidneys or cardiomyocytes, so there is no cell culture model available, and you would then probably have another alternative, another idea how to use SILAC. And of course, this is also what I did in the past, is so that we might then think about to SILAC label a living animal, right? In that case, we selected the mouse because this is probably the most commonly used organism or model organism to study all these human diseases, or some human diseases. And of course, I mean, since the last years, time is flying, a lot of model organisms were labeled. So as I said, I mean, this SILAC or this stable isotope labeling idea was not really new in 2002, right? Although SILAC was invented in 2002, already almost 100 years ago, people worked already on labeling even living animals. Of course, they don't have a mass spec as a readout, but they work to trace living animals with radioactive tracers and label them. Yeah? And of course, in our days, so now in 2008, the SILAC mouse was invented, similar to the N15 rat. And from that time onwards, a lot of other model organisms like the fly, so the Drosophila, zebrafish, SILAC, Hydra even, and Daphnia were labeled with stable isotopes. Yeah? So as a model to investigate protein dynamics and protein changes. Yeah? And of course also C. elegans. And today I want to stress a bit the SILAC mouse, since this might be one of the most important model organisms. So how we did that? I mean, of course, the mice were not drinking media, the heavy media, so they have to eat something, right? And the idea was to use a protein-free diet, yeah, and spike in our heavy lysine as an amino acid. We compress because they like these chips or these pellets, right? And then you can basically purchase those heavy labeled food from a certain company, obviously Silantes here. Huh? Okay, so, and what basically we did at the beginning, we started to label our mice with this diet here for 14 days, just to check how the incorporation takes place. And then we looked for the incorporation of the heavy lysine. So this might be the light peak, this is the heavy peak, right? And now what we did is we feed up those animals for a couple of weeks. So then we made them with a non-labeled male, right? So it's not necessary to label the male because the contribution of the male for the next generation is quite low. Yeah, so meaning the pregnant woman is then always feeding up with Elidin 6, and then the pups, if they grow up, so they are then fully labeled, right? So of course you always have to maintain the heavy diet, and again we will always maintain the females, yeah, and then you go up for the next generation and feeding them always with lysine 6, yeah. So meaning you have always a small population of mice which are fed up with lysine 6 to get labeled tissue from those animals. So, and then how we use the SILAC mouse, of course, I mean, what we do is we have labeled a black 6 mouse here, right? So, and this is a bit different from the typical cell culture SILAC experiment. So what we use is we have fully labeled the wild type mice here, and then we use the different tissues like the brain or the spleen or the lung or the liver, and then we could then, in that case, we use the heart here. So we isolate the heart from the heavy mouse, and then we basically used the non-labeled wild-type mice or the non-labeled knockout mice, right? And again, so this is the black mouse, this is the white mouse. There are differences for sure. However, we use it as a spike in, right? So we're always comparing these two peaks here, the light and the heavy, from the control mouse, and the light and the heavy peak from the knockout mice, right? And by dividing these two ratios to each other, So label against control, label against knockout. Let's cancel out the label, and whatever this label is, we have a direct comparison of the control and the knockout. So that's basically the idea, to use these living animals as a source to basically, these labeled SILAC animals as a source for a quantification spike in, basically. Yeah? And again, so what we do is we do two SILAC experiments, right? So we spike it in the control, and we spike it in the experimental condition, and then we quantify the peptides. Yeah? Good. So this allows us a very accurate quantification compared to label-free. Yeah? Good. So, however, I mean, this was a nice technique. I mean, we used it a lot, and also in our days, we use it a lot to quantify and to estimate the PTM enrichments. However, so another topic to using the lysine 6 labeled mouse is basically to study protein turnover. Yeah, I mean, what you could imagine is you have your light amino acids here, right? And these protein are then, or these amino acids are used for protein synthesis. Of course, they are then again degraded and reaching the amino acid pool, or they are completely metabolized. Yeah? So that's the idea, how you basically have the homeostasis of proteins. Yeah? So a balance between protein synthesis and protein degradation. And what we could do is, of course, we could, instead of using the lysine 6 here, which is non-labeled, we could spike in our lysine 6 as we did before, right? So to see what kind of proteins are newly synthesized in a certain time frame. And that is basically also used in cell culture. So these pulse SILAC experiments are quite popular nowadays. A lot of people using them to see protein dynamics and protein synthesis and degradation over time. And we thought, why not using the lysine 6 and spike it in for a different time in living animals, and then see how proteins are newly synthesized. And this is just one example here, right? So we used basically the unlabeled mouse. We have our heavy diet. We feed it the animals for 4 weeks, right? And then we basically see that the weight, I mean, this is what we did as to control the weight, whether the diet somehow influences the mouse behavior. So there's no obvious behavior, no weight loss, everything is the same compared to mice which are under regular diet. And we did it not only 3 times, but several times. And then, of course, what we did is the easiest thing not to kill the mice. So we take some blood samples, right? And this is serum albumin, to a very abundant protein, or the most abundant protein in blood. And also hemoglobin is quite abundant, right? Which comes from the erythrocytes. And what we see is, after 1 week already, we see a reasonable level. So this is the heavy peak, this is the light peak, right? And after 4 weeks, we have an almost complete turnover of the albumin in the mouse here, right? So everything is newly synthesized. Only a small proportion of the heavy albumin is left over, whereas the hemoglobin seems to be much more stable, right? It looks like that. However, I mean, the albumin comes from the liver cells, right? In the synthesized there. The hemoglobin is in the erythrocytes, right? So meaning we have just the half-life of the erythrocyte, what we see here for the hemoglobin. Yeah. So one has to take care which tissue and which cell type you analyze, yeah, because these turnovers not only basically associated to its endogenous turnover or stability of the protein, but also to the half-life of the cell. So if the cells will divide, yeah, or will be degraded or depleted, then of course you will also lose that protein, yeah, and might get then another turnover.
So,
okay, this
Markus,
is
what
yeah.
If I may quickly interrupt you, we have a question.
Yeah, sure.
Thanks for your talk. How do you handle the loss in coverage induced by using SILAC or other multiplexing methods?
Of course, I mean, that's again not a problem, but it's a valid point. I mean, that we reduce basically, or we increase the complexity of our samples, right? So we have more, I mean, since we have a light and heavy peak, right? So we have to basically sequence both of them. So we get a reduced number, overall reduced number of proteins. Sorry, and of course, I mean, if you don't find, I mean, for the SILAC mouse, right? This is of course true. So what we always have to look for the pairs in the light control versus the heavy spike in and versus the experimental and heavy spike in, we always should find 2 pairs, right? If we don't find them, we usually don't use them, right? So meaning overall, indeed, and that's how it is, we get less identifications compared to a label-free protein quantification. So this is basically kind of a disadvantage if you would like so. However, those proteins which you quantify, they are quite accurate and robust identified. I don't know whether this answers the question, or I mean, this guy could also say something now. I mean, whatever.
Thanks a lot. Thanks a lot.
Okay, we might discuss it later again, right? Okay, so now, but this is definitely a drawback, and therefore people like this label-free quantification, right? Nevertheless, so coming back to this turnover here, so what we see is basically different incorporation rates in different tissues, right? However, I highlighted a bit the serum albumin, and if you now look, okay, sorry, I forgot to explain that. So what we have here is in this bar plotting here, we have the SILAC ratios, right? So 1 means 1:1, 50% labeled, and this is heavily labeled, right? As I said, after 4 weeks, we have a heavy to light ratio of 10. So we have 10 times more, if you want to like that, as serum albumin compared to the light situation. Yeah, and we have always very similar ratios for the serum albumin, whether we look in heart, in gut epithelia, in liver, in platelets, or in other cells. So the serum albumin is very, very similar. So, of course, if you know, would say that serum albumin comes from the liver, it makes sense that this is basically a kind of a contamination in those tissues which also contains blood. Yeah, so meaning this is always originating from serum albumin. Yeah, this thing. And of course, now if you want to compare the half-life of the alpha enolase in heart and gut epithelium, you get completely different incorporation rates, right? Here you have a heavily labeled because the gut epithelium has also kind of a proliferation rate every 3 to 4 weeks, whereas the heart is mainly postmitotic. So here we don't have, in cardiomyocytes at least, no proliferation, or really a minor proliferation or no proliferation. Yeah, and of course the same is true for the liver, where we have a much higher incorporation compared to the heart. Yeah, and that basically allows you to study those incorporation of different tissues. Yeah, but of course, the comparison of different proteins is, of course, then difficult. And therefore, we decided a bit to study a bit more the half-life of specific proteins in cardiomyocytes. So what we did is, and since the heart is also not containing only cardiomyocytes, but also other cells like fibroblasts, endothelial cells, and probably other immune cells. So what we did is we isolated postmitotic cardiomyocytes from an adult mouse, right, which we have fed for 1 to 6 weeks with lysine 6, right? And then, of course, we isolate after every week the cardiomyocyte and also the brain, which I will not talk about today. And we might then get incorporation of lysine 6 and see which proteins are faster labeled or more labeled compared to those which are less labeled. And that is what we see again. So we counted basically the proteins here in this bins, which were labeled for different incorporation. So this is now a relative labeling. Here 0 means no labeling, 1 means 100% labeling. And what we see is that most of the proteins after 1 week in cardiomyocyte are labeled between 20 to 50%, right? So this is the number of proteins per bin. Yeah, and now, of course, if we increase the labeling for 2 weeks and 3 weeks, we see a shift up to whatever 6 weeks. And here we see that most of the heart proteins are to 80 to 90% turnover. Whereas, of course, those proteins like histones and lamine and so on, nuclear proteins, so they are only to 10 to 20% labeled. Yeah? So that's what we see if we now look in postnatal adult cardiomyocytes, which were labeled for 1 to 6 weeks. So, of course, this is nice to know that we have this increase in incorporation, and we could also basically do the same thing as a kind of a line plot here, right? So these are the same data as I showed you before, but here we just took the incorporation from 0 to 1% and the time, yeah, and that is the incorporation what we do. Of course, and what we like to use is to calculate the turnover, because this is just the incorporation of lysine 6, and it is not so easy to calculate from this incorporation into turnover, because it's not a linear thing here. You see this sigmoidal shape, right? It's not an exponential curve, yeah, but it's more like a delayed incorporation. And that's one reason for that is basically the recycling of amino acids over these 6 weeks, right? So whenever a protein is degraded, a light protein and a heavy protein, so then it's feeding up back to 80% to the amino acid and to the protein turnover, and that basically gives us this delay here. However, there were quite some nice studies from different labs here shown in this publication, which models system or which develops mathematical models, how we can use this incorporation rates here over time to calculate the true turnover, so the half-life of proteins. I will not go into detail about that today, but we basically did that and we used these tools, different tools, and what we see now is we can use, based on this incorporation rates, we can then see the half-life in days. So these numbers, what you see here, is the half-life of proteins in cardiomyocytes, right? And it ranges from 1 to 2 days up to 20 to 30 days. Yeah, so that's basically what we see in the cardiac proteome. In terms of turnover. So this might be also a valid situation for your experiment, whatever cell types you have. Of course, if you have proliferating cells, you should take, of course, a proliferation rate into account, which is also possible. Okay, so then, I mean, this is what we did with the cardiomyocytes. However, we did the same things also with muscle tissue, with skeletal muscle tissue, right? And here are some examples how we monitor, again, this is enabling here, from 0 to 100%, and here it goes up from 7 to 28 days. Yeah? So that's what we see for these different myosines here or for mitochondrial proteins. And then to summarize it a bit, so the idea was now, and this is also known in the literature and was shown a couple of years ago, that slow muscles, so we have different muscle types, right? So this is the soleus muscle or the gastrocnemius muscle. So we have different muscles, and either this is, for example, a slow muscle, and the EDL is a fast muscle. Yeah, so you could imagine marathon runners have rather slow muscles. I mean, they run quite fast, but these muscles can basically last not forever, but for a long time. Whereas sprinters, so they might basically more develop their fast muscle fibers, yeah, like the EDL muscle. And the idea was, and this was shown by other people with other technique, is that fast muscles have a slower turnover compared to slow muscles. Yeah, so slow muscles, and this is the slow muscle here, the incorporation, and this is the density or the concentration. So they have basically a faster turnover compared to fast muscles. Yeah, I mean, this is the fast muscle here, sorry, the red one, this is the EDL and the gastrocnemicus, and the blue muscles, which shows a faster turnover, a quicker labeling over time, and this basically that's what we want to show as a kind of a control experiment. And of course, you can not only see the general trend about the incorporation over time in muscle tissue, however, we could also then look for different proteins, for individual proteins. And as I said, I mean, these are the different muscle fiber types here. And then we could then use, I mean, yeah, measure here's again iodine as a contaminant from the liver, right? So this is a quite high labeling after 4 weeks or 40 days, sorry, after 40 days, whereas other proteins are much less labeled, right? And we see also difference between different fiber types. Yeah, so those proteins in the soleus muscle, so they are much higher labeled compared to the same proteins in the EDL or gastrocnemicus. Yeah? So that's basically the idea behind that, to show also the incorporation in different tissues and different muscle fibers. And of course, what we also did is we looked a bit closer into the mitochondrial proteome, and then we were quite happy to see that these are the different complexes, right? I mean, this is a mitochondria in a membrane here, and you see the complex 1, complex 2, complex 3, 4, and 5. So this is the oxidative phosphorylation to generate ATP for energy consumption. And here what we see is for the Q and N module, which are close to the matrix and to the reactive oxygen species, which shows up here. So these have a higher turnover compared to those proteins which are hidden and protected in the membrane, right? So that was also quite nice because we measured everything and then we aligned those subunits of the complex 1. And what we see is that those proteins which are located at the basically matrix site, so they show in tendency a higher labeling, meaning a higher turnover, so that they're exchanged because they might be damaged and degraded over time, yeah, and then of course newly synthesized, whereas those proteins are quite stable here, and of course the assembly factors which build up these complexes, they show also a quite high turnover. Okay, and here we also compared the turnover of those COX complexes and proteins between skeletal muscle, liver, heart, and brain. And we see that the muscle seems to be the highest turnover compared to the other tissues. Yeah. Okay, this is just to also show you that this lysine 6 labeling might also be able to verify and to decipher the different subunits of a certain complex, whether they are equally labeled or there are differences, in terms of assembly and in terms of turnover. Okay, so coming now to the last part, what I want to show you is the so-called SILAC flooding dose approach. I mean, this is another option what you could do in terms of turnover. So in the first part, we just simply feeding up the animals for a certain time period, and then you might harvest and collect the tissues or the blood. However, what we thought is we have our completely labeled SILAC mouse, and what we then did is we isolated the blood. So we isolated SILAC 0 in that case. And then what we did is we injected roughly 300 microliter into a non-labeled mouse. So that is quite fine for the mouse. There's no problem with that. And of course, they don't like to be injected with something. But so we injected 300 microliter of the heavy blood into this mouse. And why we do that? So the idea was now to monitor the heavy peak in the blood after a certain time point. So time point 1, time point 2, and time point 3. And this allows us to say or to calculate how long a blood or protein is present in the blood. And that gives us the half-life of the blood proteins in the serum or in plasma. And of course, so then that's the theory behind that. So what we then see is that we have a pool, a spike in bolus of these orange peaks here, and then of course over time we see measurement, we see one concentration, and then we measure a decay, and this is basically the clearance here is basically looks like a radioactive decay, right? So this is basically the exponential decay of those proteins which are spiked into the blood. And of course, that's what we measure here. So we calculated the heavy fraction with this formula here, just by dividing the measured heavy to light ratios here to get the decay of these proteins over time, right? And this resembles this exponential equation here. And this is the time after 3, 6, 10, and 12 hours. So relatively short after the injection. And then, of course, you can logarithmize this relative labeling here to get this formula here, right? And then at the end, we then fit the linear regression to this log values here. And then, based on this formula here, with the radioactive decay or with the half-life calculation, we could then calculate the half-life of these proteins in the blood. So that's the idea. It's not a complete half-life of the protein because these proteins are synthesized in the liver and then secreted into the blood system. So what we measure here is the time, how long these proteins stay in the blood. And then what we did it not, of course, I mean, we did it several times. And here just some spectra, which you basically might get after different time points, after 100 minutes, 720 minutes. And here, and then what we see is if we now calculate the decay or the slope of this decay, so this basically gives us the half-life at the end. So here for this protein, for the apolipoprotein, we have a half-life of 11 hours. And for another protein, in that case, the IgG, we have a half-life of 50 hours or 45 hours. And of course, we have not only these two proteins, we have also then measured some other proteins, so which allows us basically to monitor the half-life of those different proteins in the blood. So this is always a time here, which ranges from down to 5 minutes to 5 hours, to up to 70 hours. And this allows us to monitor the activity of the blood. And of course, this was just wild-type mouse here. But of course, what we also did then to prove the system, we used the so-called beta-microglobulin deficient animals. And this beta-microglobulin is localized in this major histocompatibility complex, class 1 complex. That's a heteromere containing of the beta-microglobulin and the neonatal Fc receptor here. And this complex is, besides other functions, basically quite important for the uptake of IgG. And if you don't have these beta-microglobulin, the receptor has a lower affinity to the IgGs, and those are then at the end less stable. So meaning what we would expect, and this was also shown by this study here, that those beta microglobulin mice, they should have a reduced half-life of their IgGs. Yeah? And we thought, why not? We had access to these mice at that time. And then what we did is we spiked in these blood bolus from the lysin 6 heavy wild-type mice, and the same to the beta microglobulin mice. So we did it 4 or 5 times. And then we calculated basically the different concentration of the beta, of the IgGs over time, right? And of course, we proved also whether the beta-2 microglobulin is really ablated. So by using a normal spike-in as a heavy spike-in, what we see here, this is the spike-in of the beta-2 microglobulin from the heavy mouse, and we don't see any beta microglobulin in the knockout, whereas the wild-type have some, right? Okay, so then of course we calculated the half-life of the IgGs in the wild-type and beta-2 microglobulin mice, and what we see overall a trend that these IgG have a reduced half-life in these beta-2 microglobulin mice here, these 3 IgGs, and we have some other cases. Okay, and that was also published in a couple of years ago in Analytical Chemistry and by Hendrik Neuten. Okay, so this was just another example how to use SILAC and completely labeled tissue as a spike-in to monitor the half-life of blood proteins. Yeah, and yeah, the last experiment. Do I have time? Or is it okay with the time management here? I don't know. I'm not so sure, but I hope. So what we can. Yeah,
please,
okay,
please
fine.
go ahead, Marcus.
Okay. So what we can also do is, and that was kind of a, I mean, I don't know. I mean, I wasn't so sure to do that experiment, but we were approached from another group, and they said, yeah, we need SILAC in our animals. So I mean, what I should say or explain is that the neural development in mice starts basically after birth, right? Of course, the brain is there once the mice are born, but the differentiation in the main changes takes place the first 2 weeks of life, yeah, in a mouse. And of course, a lot of people are interested in this early neurogenesis, right? To monitor and to study the early neurogenesis in mice. Yeah? And the problem is of course, these mice will not eat something. So they drink milk the first 2 weeks. Yeah. So we don't have any, and this is basically in that case not possible to label those animals. I mean, we thought to. Yeah? However, so what we then did is we used our non-labeled mice, and we basically take these mice away from this unlabeled mother, right? And then after 2 days, and then we switched to a completely SILAC mouse, which were also pregnant at the same time, to this mother here, right? So meaning we make a kind of a switch of embryos, yeah? So we took away the embryos from the SILAC mouse and give them or give her the non-labeled mice after 2 days of birth, right? So meaning we get a breastfeeding with this lysin 6 containing milk, yeah, for 1 week in that case or even longer, yeah. And then of course we measured the incorporation of this labeled embryos here or newborns from day 2 to day 10, right? So this is 1 week or 8 days labeling, and that's what we see here in terms of labeling. So basically it works, right? We get an incorporation of 20 to 30% of most of the brain proteins, and we also see labeling in heart, and reflects the adult situation in the liver. We get even more labeling. Yeah, so now we basically were quite happy that this worked, and then together with Carmen Birchmeier from the MDC in Berlin at that time, we basically investigated the NRG1 and ERP3 knockout animals and studied basically the myelation and the development of the brain during that time. Yeah, so that was quite successful. There's another example how to use the SILAC mouse. Of course, this is much an effort. So there are other examples where we treated pregnant mothers to SILAC label the beta cells, but I will not touch it today. Okay, just as a kind of a short summary, I mean, of course, what you can do is, of course, you can use these heavy food to label your SILAC mouse, right? And then what also, I mean, to invent this SILAC zoo, you might then use these other yeast or SILAC bacteria to feed up the yeast worm, the SILAC fly, or even the Daphnia if interested, and these were eaten then by the Hydra. Of course, this is kind of a niche project, I would say. Nevertheless, so these were invented in the past and also shown here by these publications. And even the SILAC mouse liver were used to feed up the SILAC planaria get a turnover or to get a kind of an incorporation of heavy isotopes, and the newt, which has a high capacity for tissue regeneration, and we use that. Okay, and of course the fish can also be labeled. So that's basically kind of a SILAC food chain to invent and to introduce these stable isotopes into those living animals. And just as a second summary, and this is, I mean, of course, I can summarize a lot, but I just want to stress and highlight again the power of SILAC, right? So metabolic labeling, and label-free labeling, right? So the advantage is if you have cells or tissues, you can immediately after protein extraction, immediately mix up your samples together. And whatever happens then in terms of enrichment, purification, or fractionation, so this happens together. And even each mistake, what you do here is the same, because you have just one Eppendorf tube. And then, of course, this gives you much more robust data. However, if you do the label-free quantification, you always have to go for this independent rows. And this might then also introduce some errors in terms of accuracy and variation. Okay, so with that, I would basically thank, of course, my team, and of course, previous members and collaborators here, and of course, all the funding, and of course, also Silantes, which always helps us with the isotopes, and just make some commercials. In case you are interested, open postdoc and PhD positions are available in Cologne, so just ask me or just approach me by email. Okay, and thanks for your attention, and open for questions.
Thank you very much, Markus. It was a very interesting presentation. So for our attendees, you are welcome to raise your hand if you want, and I can activate your microphone and your video so you can directly post your question to Markus, or if you want, you can use the chat function.
Stop the sharing, or I can edit and we can discuss it again.
So you can leave it on if you want.
Yeah.
So we have a question. Let me allow your microphone and your camera. So you can now yourself unmute yourself. John David Henderson.
Yeah. Hello. You can hear me? Okay.
Yeah. Hi.
Yes.
No, I can also see you. Yeah. Cool. Just
Great.
to see another face. I mean, Martin is a nice guy, but just to see somebody else, right?
Thank you. Cool. Thank you for the really fantastic presentation there. I just have a really basic question. So you just the data you've all shown there has always been using 13C lysine. I mean, is there an advantage compared to using like 15N nitrogen? What's the difference between the two? I mean, it would seem to me that it might be a bit more expensive using the to create chow with the lysine 6 inside it. So I just want to know if you compared these two different strategies, 15N and 13C.
Yeah, I mean, of course, I mean, there is also the 15N labeling, right? I mean, the red was labeled as 15N, and of course, at that day, there was a great competition between the Yates lab and our lab, of course, and so the point, I haven't stressed it today because, I mean, anyhow, it was too long. So the point is, N50 labeling is definitely more difficult, right? Because what we do with SILAC amino acids, we just label the lysine in that case, right? So you might also ask, why not arginine? So arginine is in cell culture an essential amino acid, but not in living animals. So arginine is heavily converted. So therefore, we just go for lysine, which is also bad in terms of trypsin digestion, right? We cannot use trypsin, so we use lysine for the digestion, to get only lysine-containing peptides. And the advantage is we just play with the lysine, and we have always the same distance. So whatever we do, lysine 6, for example, right, has a certain pair, which is also nice for Max Planck, by the way, and Jürgen might also say something on that. If you label N15, which has also advantages in terms of metabolomics, and Christoph Todt next week we might also stress that point. But if you label your proteome with N15, you have to label all the nitrogen atoms, right? Which gives you much more complex spectra. And of course, the distance between the light and the heavy is always different because it depends on the number of nitrogen atoms in your peptide. A short peptide or longer peptide, or depends how much nitrogen atoms you have in your sample, gives you different distances, right? And also the spectra, the isotopic envelope is much more complicated, right? And a small contamination will also basically reduce the accuracy, yeah, of the quantification. So meaning, I would say, I mean, both methods are good. I mean, you can use both ways. However, I would say SILAC labeling is much easier in terms of cost. No idea. I mean, that's what you should discuss with Silantes, right? They have, I think, both options. And but so my impression is that SILAC is much easier. But again, I mean, lyc digestion is a limitation factor. The overall number is, and lyc is nowadays quite expensive. But I saw a lot of companies nowadays instead of this one company. So now they're producing lyc, and you might get it much cheaper now next couple of months, hopefully. And then, yeah, then that might be an option. Yeah. So, but if you want to go for, let's say, metabolites, right? How then nitrogen is converted into smaller molecules and so on. So then N15 might be better because that's what you do not see for lysine 6. Yeah? No, lysine 6 is also degraded and might be introduced into these creatine pathway, but it is, but not to other proteins. Yeah? And therefore, I think this is more like a robust method compared to N50 labeling. But if you ask other guys, they might answer differently.
That was great. Thank you. Such a comprehensive answer. Thank you so much.
Yeah, good. Yeah, thanks for the question.
Thank you, Dr. Henderson, for your question. So, any other questions? Yes.
I know this guy.
Hermann, I'm allowing your camera and your microphone. You can now unmute yourself. Hermann, you need to unmute yourself. You can do that in your top panel with the microphone and the camera. Not yet. We can't hear you yet.
Can you not unmute him by yourself? Can you not do that?
Let me try that.
Okay. Now I have.
No,
okay.
But I can't see you. I mean, but you might not change. I know how you look like, but I don't see your camera.
There is also next to the microphone.
The microphone. The microphone is on.
No, I see you. Yeah.
No,
you haven't changed. You haven't changed. So then.
Okay. Yeah, good to see you. We had some collaboration also on the common patent in the food chain stuff, and I think again it's always nice to collaborate with you. And thank you very much for presenting the seminar, which showed very clearly the potential of the SILAC approach. For me, somehow new was, because I'm not anymore so much in the field, is the flooding. So that's a really nice experiment which you can do with the flooding approach. A kind of, let's say, type of SILAC, a special application. And I think in principle you can apply it to all body liquids. I think that is a prerequisite that's liquid somehow. Otherwise, you can do that.
CSF, basically, you can use CSF, urine. I mean, of course, it should be somehow bring back to the mice, right? I mean, in the mice, you have, again, I mean, blood is, of course, a main system. So CSF might be something to take out from the mouse, from the mouse.
Right. Yeah. Well, my question was a physiological question. Blood is not blood. Blood is free running. Blood is in all the different tissues and might have different lifetimes over there. How do you deal with that?
So in that study, we haven't looked for the basically taking up blood proteins, right, in other tissues. Yeah, I mean, but as I said, I mean, when we look for the other tissues, right, I mean, so in the past, just let's say when we look for the muscle, right, and then we see albumin. Okay, then we said, okay, albumin, of course, the muscle has a lot of blood, right? Even if we flush the blood out, there might be still remnants and so on. Yeah. So, but what we do now is we're doing the same labeling experiments over time with single muscle fibers. So what we do is we isolate the muscle, flush it, of course. We digest the muscle with collagenase, so everything is digested outside the cell. We isolate it in a single fiber, yeah, and then we measure the proteome. And what we always see is we see albumin in those fibers. We have no idea why, but we see albumin. And this albumin has the same incorporation rate compared to the liver. So meaning it is taken up by the muscle cell, right? And this is also known from other studies, but we haven't really looked how long is it then there and what happens with that over time, because this is a bit challenging, yeah, I mean, to see that. But I mean, here in this SILAC flooding approach, we just looked again in the blood and not so much about the uptake. Of course, at a certain time, those proteins are taken up by other organs and tissues, but this might be also a bit challenging actually to study that, right? Because then you should basically stop the labeling and then see what happened with those proteins, right? So this might be an option, right? This might be a cool experiment to see the labeled albumin in muscle, which is taken up, and then stop the labeling and see how long it will basically be there in the muscle. To see what happens with albumin. Because if you constantly feed up, then you get new albumin in the muscle, right? And new albumin in the muscle. So this makes it difficult to distinguish. But if you then make a bolus and then, as I said, I mean, and then you look, yeah, this might be something, yeah.
Okay.
Yeah.
Okay. Yeah. Good. Thanks.
Cool. Yeah. Good.
Thank you, Hermann.
I think we are a bit late, right? Jürgen is probably waiting, yeah? But
yeah, I can quickly announce in parallel, we have already started with our next webinar. So general information, our webinars are all on video. So you can also watch the webinars that you missed on the Silantes website afterwards. So please, if you have further questions, feel free to ask them now. Otherwise, Markus, I want to thank you very much. It was a very interesting and informative talk. And we really appreciate your help and your time. And I hope to see you soon in person again.
Okay, yeah. Thanks a lot for everything. And yeah, enjoy the afternoon, enjoy the other session, right? Okay.
Thank you very much. Have a good day.
Bye. Bye-bye.